Hi, in this video, I'll cover how to use MrBean for spatial analysis of field trials with splines, or SPATs. To start, we'll launch the app from RStudio with library MrBean and MrBean: run app. In the last video I showed you how to upload data into MrBean, so make sure you watch that one if you have questions about that step of the process. I'll be using the example data built into MrBean for this tutorial. To start, click on the single site dropdown and select Model Specs. Then, we can set up a single-site spatial analysis the way we wish. First, in the SPATs box, set the response variable for the analysis in the response variable dropdown box. Below that, select the column from your data corresponding to genotype IDs in the genotype box. Then, we have an option to treat this factor as a fixed effect if we uncheck the random genotype checkbox below the dropdown. This decision depends on the user and the objective of the analysis. Below this, in the box labeled checks, we can set which lines, if any, are checks, to separate those lines from the random part of the model. In the next box, labeled coordinates, we select which columns in our data refer to the layout of our experiment, with settings for column and row, as well as an option for setting replicates. In the next box, factors, you can set additional qualitative variables you wish to include in the model, either as fixed or random effects. Below this, there's a box to set covariates in the model, if you wish. In the next box, Model, we can run the model with the green Run Model button, as well as display info boxes about heritability and other information, and a 3D spatial trend with the other boxes. The spatial trend will appear near the bottom, and if the box doesn't appear right away, you can expand the box labeled Spatial Trend with the plus button on the right side of the header bar. We can also display a table of the BLUPs with the BLUPs/blues button, and download the table with the green Download Predictions button. You can also see a table of coefficients for the model with the Coefficients button, and save the table with the green Download Coefficients button. In the next box, Segments and Residuals, we can set the tolerance for outliers in the dataset with the R-limit slider, and choose to remove outliers if they fall outside the threshold with the Remove Outliers checkbox. We can see which lines are detected as potential outliers in the Residuals tab under the single site dropdown, which we can also access by clicking the green Residuals link in the Model box on the Model Specs page. Then the outliers appear in red in the plot. We can view the same BLUPs/blues table as earlier under the BLUP/blues page in the single site dropdown on the right. On this page we can also see a histogram of the predicted values and a plot of the predicted values, if we maximize the predictions plot box. In MrBean, we can also run the SPATs analysis as site-by-site instead of just for a single site. To start, click the site-by-site dropdown on the left side of the menu and select Model Specs. On this page, we can configure the analysis in a similar way as in the single-site analysis. Starting in the configuration box, we can set which columns in our data correspond to our response variables and to genotypes, and we can select which entries in the genotype column are checks with the checks box. Below this, we can set the column that contains the experiment ID, and select the columns corresponding to column and row below that. Then, click the green check button to move on to the next step. I'll be using an example dataset that I have on my computer for this part. In the next box that appears, Additional Components, we can set some additional configuration for the analysis. First, set the column corresponding to replicates in the replicate selection box. Below this, we have options to select which factors in our data are fixed factors, random factors, or covariates. Below this, like in the single-site analysis, we have an R-limit slider to set the threshold for outliers, as well as an option to remove outliers beyond this threshold from our dataset. Once we have the second configuration box set up the way we want, click the green Continue button. Then, in the Genotype Statistics box that appears, there's a chart of the number of genotypes in each experiment, split by the factor we set in the experiment box. We can also make a graph of shared genotypes between experiments with the Shared Genotypes toggle switch. Once we are ready to run the mixed model analysis on our data, click the orange Run Models button. Then, to view the results of the analysis, click the green Check Models link or select Results under the site-by-site dropdown. Then we can view several different plots, including a comparison of residual and genotypic variance by experiment, a correlation matrix of correlations between the experiments, and a plot of the genotypes ranked by the predictive values. In the upper right-hand area, we have plots of the raw data, fitted data, and residuals, as well as a plot illustrating the spatial trend of the layout. There's also a plot of the BLUPs over the layout and a histogram of the BLUPs. We can select which experiments to depict in the plots with the experiment drop-down box here, and see a summary of the analysis with the orange summary button. Also on this page, we can view and download tables containing a summary of the analysis of each experiment and the predictions of each entry. If there are any outliers detected, they will appear under the Potential Outliers tab. In MrBean, we can also run the SPATS analysis as trait by trait. To begin, click the Trait by Trait drop-down on the sidebar on the left and select Model Specs. Then, we have a configuration box similar to what is in the site-by-site analysis, with boxes to select which columns correspond to genotype, column, row, and replicate. In the first box, Response Variables, we select which columns correspond to traits we want to compare in the analysis. We can switch the Additional Components toggle switch to set factors as fixed or random effects, or covariates. Below this, we have an R-limit setting and Remove Outliers checkbox to remove outliers from our data, if desired. Once we have configured this analysis, click the green Run Models button. Then, on the right side of the page, we have the same kinds of spatial plots as appear in the site-by-site analysis. Plots of raw data, fitted data, residuals, a fitted spatial trend, and BLUPs, as well as a histogram of the BLUPs. We can select which traits analysis is plotted with the Trait Select box below the plots. On the next tab in this box, Correlations, we can see a correlation matrix of the traits we selected as response variables. Below the plot, we have buttons to show a principal component analysis or a dendrogram of our data, based on the traits we have selected. The next tab in this box, Summary, shows a table with the summary of the analyses for each trait we have selected. You can save this table with the green Download Table button. The next tab, Predictions, shows the predicted values and standard errors of each line in our dataset, which can also be saved with the Download Table button. The last tab, Potential Outliers, shows any entries that fall outside our tolerance set by the R-limit slider, if they exist. Below this, we have a plot of the genotypes ranked by predicted values, based on the trait selected in the Spatial Plot tab.